So contemplating the beginning of mankind leads us to thoughts on the origin of life itself. For this, we call on Professor Jack Szostak, Nobel laureate of Physiology or Medicine and professor in the Department of Chemistry, University of Chicago. Professor Szostak will speak to us on new perspectives on how the molecules of life were synthesized on early Earth, eventually giving rise to the varieties of life we see around us today. To achieve such innovative and pioneering research requires the willingness to venture beyond recognized boundaries of unknown territory, often outside established convention, which may challenge what might have been previously accepted as familiar terrain. Professor Szostak will describe how overcoming conceptual barriers has led to new thinking on life's origin. We'll now hear from Professor Szostak on the origins of life, not as hard as it looks. Welcome.
Okay, I'd like to begin by thanking Laurie and Bengt for inviting me back to speak in this wonderful symposium. It's a hard act to follow Don, who's given us an amazing overview of the origins of our species. But what I'd like to do is go back about another 1,000-fold in time to somewhere around 4 billion years ago, when the chemistry of the early Earth was developing and becoming more complex and ultimately giving rise to biology. So that's a hard thing to study for many reasons, including the fact that we have no actual, geological samples of the surface of our planet back then. So a lot of what I'm going to talk about is a mix of sort of theory and experiments that gives us ideas about how things might have happened. What we can do is test our ideas. We can figure out what things don't work, and what we're left with, we hope, is a possible path leading from chemistry to biology. So when I was thinking about how to present all of this, one thing that really struck me, something that's happened again and again, is that we see at some point an advance in the field, which usually results from overcoming some preconception, some deeply ingrained idea about how things ought to work, which turns out not to be true. So it's overcoming those deeply ingrained prejudices, which is really essential for the progress of science. And what I'd like to do is give you a series of advances from my field, which is the chemistry of the origins of life. This whole field is actually closely related, surprisingly, to a big part of astronomy, which is the discovery of other solar systems, exoplanets. We now know that there are millions, probably billions of other planets, many of which are like the Earth, orbiting other stars. And that's given rise to a huge hunt for signs of life out there. I'd just like to ask everybody in the audience here, how many of you really think that there is life out there on other planets? Just raise your hands if you think there's life out there. Okay, and how many of you are equally sure that we're the only place that has life? And I see there are a lot of people like that too. So we have these very divergent views, right? Life you know, we've seen that it arose here. It's given rise to this incredible diversity that we see around us. Some people think that must happen out there. Other people think, well, life is so complicated, maybe it only got started here. If we could find another independent origin of life, that would clearly say that it's not so hard to get from chemistry to biology. But so far, we have only one example. And so my view is that we don't know, we can't say. What we're trying to do in the lab is figure out if there's what looks like an easy path to biology. But I think it's our friends in astronomy who will perhaps most clearly answer this question. Okay, let's see. Okay, all right. So, yeah, I wanted to just briefly say, why do so many of us think that life is common in the universe? So clearly one example is there are lots and lots and lots of environments that could support life, right? We have already in the early days of this adventure, over 5,000 confirmed exoplanets, planets orbiting other stars. And many of those are in the habitable zone where you could have liquid water. And so it's pretty clear that a lot of these environments out there could support life. We just don't know if they do. I think another reason for thinking that life might be common is that there was a lot of time available on our planet for life to get started. So in between the time when the Earth had cooled down after the moon-forming impact to the first clear signs of biology, there's a time span of at least several hundred million years, maybe as much as 700 million years. And so there was a lot of time for life to get started, and no matter how hard it is, maybe in the end it was inevitable. On the other hand, I think what makes a lot of us think that life might be rare and really hard to get started is that modern life is just so incredibly complicated, right? So this is part of the metabolic chart. I mean, every living organism on the planet has thousands of coordinated chemical reactions going on all the time, catalyzed by incredibly complicated and beautiful enzymes. But even the informational organization of life is complicated, right? So from the central dogma of going from information coded in DNA, archived in DNA, transcribed into RNA, used to code for proteins, which carry out all the reactions of cells. Every part of that system depends on every other part, right? It's incredibly self-referential. You can't replicate DNA without help from RNA as primers, protein enzymes that are polymerases and other parts of the machinery and so on. Same thing for RNA. It's coded generally by DNA, but to replicate it or to transcribe it requires proteins and metabolites. So how could such a self-referential system ever evolve? There are a lot of crazy theories to account for this, but things didn't get, progress wasn't really possible until some very smart people like Francis Crick, Leslie Orgel, Carl Woese realized in the 60s that it was RNA, that molecule in the middle, that potentially held the answer. When the first structures of tRNA came out with its complicated three-dimensional folded shape, these people realized that maybe RNA could also act like an enzyme, in which case you have one biopolymer that can both carry hereditary information and carry out chemical reactions. And so it became possible to at least think of an earlier, simpler stage of life's history, where before the evolution of DNA, before the evolution of proteins, you could imagine simple cells with an RNA genome that was catalyzing its own replication as well as biochemical functions. Okay, so that simplifies the problem of how life got started enormously, but we still have to work out how could the chemistry of the early planet give rise even to structures like this, which are still quite complicated. So here's a more detailed view of what we think these early simple cells might have looked like. We often call them protocells. They still have a membrane envelope that keeps things together so that different parts of the system don't just float off into space. Inside this membrane compartment, we have small bits of genetic material, which we are pretty sure was RNA or something closely similar to modern RNA, maybe not identical. These kinds of structures are actually very easy to assemble in the laboratory. But the harder part is to imagine how a system like this, a protocell, could actually grow and divide and replicate its genetic material before there was any evolved genetic material, right? We need to go through this stage where we have this self-assembling system that's complicated enough to actually undergo replication, but just based on chemistry and physics. That gives rise to the ability to begin the process of Darwinian evolution, which can lead to the complexification and diversification that we see around us now. Okay, so in thinking about how to do the replication of primitive RNA molecules, I want to begin by mentioning one of the pioneers in this field, the late Leslie Orgel, who had been working ever since the late 60s, early 70s on how to imagine replication happening without enzymes. And one of the key insights that Leslie had, out of many, was the fact that the substrates, the nucleotide molecules that would be polymerized during the process of replication, had to be different from what we see in biology today. So what you see on the top, the nucleoside triphosphates, are the universal substrates for DNA and RNA replication now, but they require very sophisticated enzymes in order to accelerate that polymerization process. And if you don't have those sophisticated catalysts, you need molecules more like what you see on the bottom, those so-called phosphorimidazolides. These are molecules that are reactive enough that they can start to polymerize all by themselves without any help from sophisticated, fancy evolved catalysts. So that kind of insight gave us our kind of standard view of how this copying chemistry ought to work. And this little video that was made by Janet Iwasa shows how we were thinking about the process. We'd imagine single strands of RNA floating around in this chemically very rich environment filled with activated nucleotides that could find their partners by Watson-Crick-Franklin base pairing and sort of click into place, reacting with the growing chain and building up a copy, as you see here. So to look at that in a little bit more detail, what you can see in the diagram is that we have a short strand at the top, a primer, which is growing by copying the template. And the way we imagine this working is based on what we know about how this happens in biology. We imagine a single activated nucleotide coming in, binding to the template, the bottom strand, next to the primer. And for those of you who are interested in the details of the chemistry, the atomic structure of one of those substrates is shown at the bottom, but the schematic version at the top shows you the essence of it. So this nucleotide comes in and binds, the primer attacks, adds one nucleotide, and kicks out the green imidazole moiety as the leaving group. And I'll show you an example of an experiment like that. This is the kind of thing that was pioneered in Leslie's lab. This is an experiment we did in my lab many years ago. And it shows the progress of this type of spontaneous copying chemistry. No enzymes involved. Okay? So in the gel image underneath the sequence diagram, at time 0 is the starting material. After a minute, you can already see a slower moving band that represents the primer where one base has been added. After a few more minutes, you see a second base coming in, and then a third base coming in by 5 to 10 minutes. So the basic chemistry of copying works, but there's a puzzle hidden in this experiment. And if you look closely at the template sequence, you'll see that there are 4 Cs, which Gs can come in and base pair, making a GC base pair. But you see in the experiment, only 3 of them get copied, right? And why is that? This was a puzzle that was in the literature for at least 40 years. And it was just one of those things that there was like no obvious explanation for. And so it kind of lay there. Remarkably, really remarkably to me, in Leslie's lab about 25 years ago, they did a very clear set of experiments that showed that when a second nucleotide binds to the primer, downstream of the one that's going to react, it accelerates the reaction. That second nucleotide is acting as a catalyst. Right? It just seemed like an incredibly important clue as to the mechanism of this process. But that discovery lay there uncited, unseen, unknown for almost 25 years until a fantastic student in my lab, Noam Privas, rediscovered this independently from his own experiments. To me, this always seems like a really iconic example of the fact that by working hard in the laboratory for several months, you can save yourselves a couple of hours in the library reading the literature. So we rediscovered this, and we really tried to get to the bottom of it, to figure out the mechanism of this catalysis. And we spent several years on this. We tried all kinds of things. Our bias was that this catalytic effect was probably some kind of molecular interaction. Some part of the second nucleotide was maybe helping to pre-organize the structure of the reactive nucleotide and to make it, in the right geometry for the reaction to go. But we could never nail that down. And finally, a new student came into the lab, Travis Walton, and he swore he would get to the bottom of this mystery, and amazingly, he did. And what he discovered was that the interaction between these 2 nucleotides, the catalyst and the one that's going to react, was not, as we thought, a non-covalent reaction involving a change in geometry, but actually due to the formation of a covalent intermediate. So the process is diagrammed on the left, and the chemical details are on the right. But basically what happens is that two of these activated nucleotides react with each other. They make a strange-looking dinucleotide that binds to the template by 2 Watson-Crick base pairs. So it can bind more tightly, which means you need less of it, which is nice. And we were able to show later from structural studies that bridge, that phosphate, imidazole phosphate bridge, is pre-organized in 3-dimensional space to favor the chemical reaction of template copying. And so I think what took so long to make this discovery was the fact that we were convinced that this non-enzymatic copying ought to work the same way as it does in biology. It just doesn't. Okay, so once we learned about that, once we got over that barrier, we were able to take advantage of our understanding of the chemistry and really learn how to copy RNA templates in a much more efficient and general way. And that work is still ongoing, but this was the key advance. It came from overcoming that preconception. Okay. Now, I haven't said anything so far about where do these nucleotides come from. I mean, these are pretty complicated molecules. Why would they be lying around on the surface of the early Earth? And in fact, 30 years ago, actually, Jerry Joyce and Leslie Orgel summarized the general feeling that the way molecular biologists were studying the problem was unrealistic, to put it mildly. Right? They called it the molecular biologist's dream of some warm little prebiotic pool filled with pure, concentrated, activated ribonucleotides. How could that possibly be? So, in fact, the chemistry of nucleotide synthesis at that time was not very well developed. And a lot of people actually came to the conclusion that maybe it was actually chemically impossible to get to RNA in one step. And maybe there was some earlier, more primitive version, some simpler genetic material that would have been easier to make and easier to copy, that then later on evolved into RNA and DNA. In the intervening, that led to a huge blossoming of studies of different kinds of nucleotides. A lot of the work done by one of the great chemists of the preceding century, Albert Eschenmoser. But in the end, nothing as good as our ribonucleotides ever came out. Still has left the problem, though, that in the chemistry going on, not in a nice, clean lab where everything is well controlled, but in a natural environment, it's not going to lead to pure ribonucleotides, right? We are now pretty sure, on the basis of a lot of chemistry that's been worked out by many people, but a lot by John Sutherland in the UK, that the synthesis of ribonucleotides is actually reasonably, not completely, but reasonably well understood. But it should be accompanied by at least some synthesis of closely related materials like arabinonucleotides, threonucleotides, and even deoxynucleotides. So any prebiotic synthesis, you'd expect to have some mixture of these different nucleotides, which could then polymerize into short chains, but they'd be kind of messy, very heterogeneous, right? And that doesn't seem like a good, very promising start to something that could easily replicate and actually fold up into structures that might begin to do interesting catalytic processes. So this is something we've been studying in my lab over the last 10 years. I'm just going to give you two quick examples that give us hints about maybe how RNA emerged as the winner from this primordial competition between different types of nucleotides. So what we can do in the lab now, and this is work, a lot of this work was done by Chris Kim, Li-Jun Zhao, and others in the lab. We look at these other possible relatives of RNA and ask, well, what about their copying chemistry? How does it compare? And if these other nucleotides were around, would it interfere with the copying of RNA? So for example, if we look at arabinonucleotides, which are very close to ribonucleotides, there's just one hydroxyl that's pointed up above the sugar ring instead of down. We can follow all the steps of the reaction, right? The activation process, the formation of that covalent dimer intermediate, the actual reaction with the primer, subsequent primer extension. What we see is the initial steps, in this case, the formation of that intermediate, it's not really affected, but the extension of a primer with an arabinonucleotide is slower than with RNA, and in a competition experiment, mostly what you see is primer extension with RNA. Not only that, but if an arabinonucleotide is incorporated into a growing chain, it's essentially a termination event. You can't extend beyond that. On the other hand, if there are arabinonucleotides in the middle of a chain, you can copy over it, but what you make is RNA. And similarly, with nucleic acids, where you have a 4-carbon sugar instead of a 5-carbon sugar, they actually look easier to make. But here, every step on the process is slower. Right? So at every step, RNA is outcompeting this very similar relative. And yet again, if you have these threonucleotides in a template strand, you can copy over it, but you're more likely to make RNA. And so this has given rise to the model that you see here. We imagine somehow a primordial environment on the surface of the early Earth where these different kinds of nucleotides have accumulated, probably mostly ribonucleotides, which make RNA, but mixed in all of these competing species. They assemble into these very heterogeneous mixed strands. But every now and then, they can base pair with each other, and little bits of primer extension happen. And when that happens, what gets made preferentially is RNA. So the idea is you keep doing this, you go through cycles, what you're going to end up with is this messy early mixture is going to get transformed into strands of almost pure homogeneous RNA, just like what we see now. Okay. All right, so we can see at least bits and pictures of the pieces of how to, from the Sutherland work, how to make nucleotides, from these experiments, how they assemble into primitive strands that are RNA. We know how to copy them. What's the next step? How can you actually get replication to work? So I need to explain why this has been such a difficult problem. In fact, it's one of those problems that was so hard, we didn't even want to think about it for many, many years, because there was like just no obvious way around it. So to explain, if you have a strand of RNA and we want to copy it, if you have enzymes, if you have all the evolved machinery of biology, it's no problem. You can start at the beginning, quickly extend, make your copy, and stop at the end. But if you don't have that evolved machinery, there are all kinds of problems. The only way to start at the beginning is with a specific primer that will bind to the template strand at the beginning. But there's no way to supply a specific primer sequence, in a prebiotic situation where you don't have catalysts or any way to make it. Even if you can copy an RNA strand, if it's above a certain length, it gets very hard to get the strands apart, even at high temperatures. It's very difficult. And furthermore, as soon as the temperature goes back down to a point where base pairing can happen and copying chemistry can happen, these complementary strands come back together thousands of times faster than the chemistry that is required to copy a template strand. So it just looks impossible when you view it this way. There are other problems as well. I already showed you an example of what we used to call the last base addition problem. Now we understand why it's so hard to add that last base, because the two nucleotide real substrate can only bind very weakly by one nucleotide. So that reaction's incredibly slow. And there are other problems. If you start in the middle, then you lose the information from the beginning, and that's going to happen on both strands. So replication looked very difficult. So we've all been through a difficult time with the COVID pandemic. For those of us who do laboratory work, it was a tough period because the labs were all shut down. We couldn't go in. We couldn't do our experiments. We had to just stay home. All we could do is stay home and think, which turned out to be a good thing in this case. We could still talk to each other, by Zoom. And so in the middle of the pandemic, through conversations with people in my lab, we came up with a theory, a model for how to do replication in a way that we think actually solves all of the problems that I just showed you. So we call this model the virtual circular genome model. The reason for that is we imagine the genome of a primordial cell being circular. That's illustrated by the green circle in the center. But there are no circular molecules. All we need to have are small pieces, short oligos, that map to a circular sequence, right, in lots of overlapping combinations from both strands. Okay, so why is that so great, you might be wondering? Well, The idea is that since you have little bits and pieces of this circular sequence from both strands, they can come together. Wherever they overlap, have complementary sequences, you have the possibility of extending those primers by the kind of primer extension chemistry that I showed you earlier. So these complementary fragments can come together in lots and lots of different ways, and some of them will allow for a bit of copying chemistry to go on. Others will not. But if you cycle the process through, you can eventually copy in little bits and pieces all the way around the circle. So by iterating that process, we think that it should actually be possible to replicate the entire circular sequence, but not like it happens in biology, where you begin at the beginning and go to the end and then stop, but in a distributed way, totally different. That then we imagine quickly evolved once you had catalytic machinery into a more efficient mechanism like we see in modern biology. Okay, so just to point out again that when these complementary pieces are allowed to anneal with each other, they can do it in lots and lots and lots of different configurations. In fact, this is one thing that makes it very hard to model this kind of process. Even for a very small sequence, there are hundreds or thousands of small subfragments. They can come together in millions of different ways. And so figuring out all of the necessary interactions is a little bit complicated, but we're working on that and collaborating with some very clever people, in particular Arvind Murugan in the physics department at UChicago is helping us try to understand this complicated system. But we think that by cycling the environment, you can repeat this process of doing little bits and pieces of copying that will eventually give rise to replication. So that's shown here. We have a cycle. Every time the environmental conditions allow these short pieces to come apart, then the conditions change to allow base pairing. They come together in different configurations that allows, in different places, little bits of copying chemistry to go on. So a lot of what we're doing in the lab now is trying to experimentally test this model and actually demonstrate replication in the lab. So we'll have to see how that goes over the next couple of years. Okay, I want to tell you another actually interesting little anecdote of where we faced a problem that really looked like it was a very hard to get over step in imagining how this copying chemistry could work. So again, the schematic of how this works is shown at the top. The chemical details are shown at the bottom of the slide. But here's the puzzle. We have these activated nucleotides. They react with each other to make this bridged dinucleotide that's the real substrate for template copying. But that reaction is reversible, okay? And the equilibrium of that reaction is actually towards the monomers, which are not good for replication. The amount of material that gets made at equilibrium that can drive copying is pretty small, okay? So it's an equilibrium process. What can you do, right? Is there any way to make this whole thing work better, to make more of the molecule that we need to do copying? And of course, there is. You know, if you think about non-equilibrium chemistry, it turns out we've actually found two ways to shift the equilibrium in the direction that we want. One is a chemical process that we actually don't really understand, so I'm not going to talk about it. The other is, well, you see the products, there are two products, right? There's the bridged material that we want, and then there's that leaving group, that little green bit, the imidazole. If we can get rid of that, we could shift the equilibrium. This is something that we're working on. We imagine this working inside protocells, membrane compartments. If we allow mononucleotides, activated nucleotides, to diffuse into this compartment, right, we'll get a little bit of the reactive dinucleotide that we want. But if we can wash out that imidazole leaving group. We can shift the equilibrium all the way towards making the actual important substrate for template copying. And that should make template copying go faster at lower concentrations, and we know from other experiments, much more accurately. So this is something we have hints that this can work. We're trying to work out a way to actually demonstrate this, which hopefully will lead to an understanding of how this could actually happen in a natural scenario. Okay, so these are the kinds of little bits of stepwise pieces, how we have to overcome one puzzle at a time to try to build up an overall picture of how to go from simple chemistry to the ability to copy and actually amplify genetic materials. Okay, so what kinds of environments on the early Earth could support chemistry like this? We need to be on the surface because a lot of the steps in nucleotide synthesis are photochemical. We need energy from the sun, from ultraviolet radiation, to drive a lot of these processes. We want an environment that we're pretty sure is not steady, but is dynamic and cycling and fluctuating all the time to drive the cycle of protocell growth and division. And so environments like that we can actually see and visit on the Earth today. Volcanically active areas like this is a picture of Yellowstone Lake in the western United States. There are many. Other regions that we think have similar kinds of surface chemistry would be in areas after a large impact on the early Earth. These environments are nice because they allow us to think about how to both make and assemble the feedstocks, the starting materials, of which, ironically, we now think that cyanide is the key ingredient for building life. Right? Cyanide is made in the atmosphere. It can come to the surface in rain, but is a very dilute solution. Right? And if you have cyanide in water at low concentration, like when it goes into the ocean, it's just going to hydrolyze by reacting with water. In order to use cyanide to build up the building blocks of life, it has to be concentrated, which has to happen on the surface. And there's a fantastic way that could happen. And this comes from insights from John Sutherland, my colleague Dimitar Sasolov, and John Grotzinger. The idea is that in a volcanic or impact crater area, you have hydrothermal circulation of water through the rocks, which brings up ions, including iron, into ponds or lakes on the surface. And iron reacts strongly and instantaneously with cyanide to make very stable complexes called ferrocyanide. And as these lakes periodically evaporate, you could build up a reservoir of cyanide held in this stable ferrocyanide intermediate. And then, again, if you're in a volcanic area, lava flows will come over this, thermally process these chemicals, and release concentrated cyanide, ammonia, cyanamide, all the reactive starting materials that we need to build up sugars, amino acids, and nucleotides. And then when they come to the surface, as rainwater leaches these things out and through the groundwater and up through springs, they can flow, for example, over, this is a terraced outflow, again in Yellowstone, be exposed to ultraviolet light, undergo chemical transformations. Intermediates, like here one of the key intermediates in the synthesis of nucleotides, tends to crystallize out. Another opportunity to build up a reservoir of a key intermediate as a crystalline reservoir. And again, contaminants, side products, byproducts can get washed away. And it's this kind of thinking about how to get away from the idea of this primordial soup where everything's just all mixed up together, right? to the new way of thinking, which is that you build up reservoirs, you undergo a few steps, make a new reservoir of the next key intermediate that can get purified. And in that way, you can build up at least an idea, a model of how these complicated transformations could actually occur in realistic geological settings. Okay. So the way we're thinking about it now, just to summarize, is that we imagine very primitive cells, protocells, whose growth and division and internal replication are driven by the environment. In modern life, of course, all these processes are internally controlled by highly evolved, sophisticated machinery, protein enzymes. But in a primitive situation, we imagine the environment is responsible for driving a cycle. And so here's one idea of how something like that could happen. You have protocells in a pond. You have hydrothermal vents releasing streams of hot water, which we see today in lakes like Yellowstone. Get caught up in a stream of hot water, these short fragments of RNA can come apart. The protocell gets swept out into cold lake water, things come together again, little bits of copying chemistry. You iterate that cycle, you can do replication, and that sets the stage for the beginnings of Darwinian evolution, which would allow for the emergence of the first RNA enzymes, which can drive better replication, allow you to have a longer genome that can encode more functions, and so on and so forth, to a more and more complicated and more well-adapted primitive cells. And eventually, over some unknown period of time, we see the evolution of metabolism, of energy transducing machinery, complicated membrane structures, the origins of protein synthesis, and so on, which sets again the stage for the evolution to the last universal common ancestor of life, which was already as complex as a modern bacterium, and then on to us. Okay, so again, just to study, I think, from all of this long history of sort of theory and laboratory experiments, one of the things that strikes me as the most important is keeping an open mind and being ready to abandon your preconceptions and think of a new way that things might actually work. And I think that's happened enough times that we're just starting now to see the outlines of a coherent pathway going all the way from the chemistry of the early Earth up to the beginnings of biology. Okay, so just wanted to mention, I've just finished writing a book with Mario Livio, who's a fantastic astronomer. And if any of you are interested in learning more about the details of that process, you can find it in this book, which will come out this fall. And so with that, I'd like to thank you all for listening. And thanks again for the opportunity, Laurie, to speak here.
Thank you.
Thanks.
Fantastic.